Part 3. Analysing the structural effects of single amino acid polymorphisms and engineering proteins for therapy. What are we trying to achieve? Well, we've seen this slide some time ago. The idea of the central dogma of biology, that DNA sequence encodes protein sequence, which folds up into protein structure, and that has a function. So we can try to understand that function from protein structure, and we can also try to predict things about the function from the sequence. And once we can understand these things, then we can hope to modulate function by intervening with drugs. So in the case of mutations, if we have a mutation in the DNA sequence, which happens to alter the protein sequence, that will result in alteration of the protein structure and therefore have some effect on function. Again, if we can understand and modulate how this happens, we can design drugs that bind to the structure, restore that structure, and therefore restore the function. So what are the aims of mutation analysis? First of all, to understand the molecular basis of the effects of mutations on protein structure and function, to be able to correlate these structural effects of mutation with clinical effects, to aid in the prediction of prognosis and guide treatment regimens, and to aid in the design of drugs to rescue protein function and give them to the right patients. Let's take p53 as an example. So p53 is a protein known as the guardian of the genome. It has a really pivotal role in the detection of DNA damage. It halts the cell cycle to allow repair enzymes to do their job, and if DNA damage is too severe, it triggers apoptosis. So if your p53 is damaged, this allows other mutations to accumulate. And it's estimated that p53 is mutated in at least 50% of human cancers, and something in the p53 pathway is almost certainly mutated in all human cancers. Interestingly, treatment is also compromised because p53 triggers apoptosis when there is considerable DNA damage. Now, traditional ways of treating cancer, such as the cytotoxic chemotherapies and things like radiotherapy, essentially work by causing massive DNA damage, and then p53 is involved in triggering apoptosis and killing those cancer cells. So if your p53 is damaged, then this process is less effective. Now, this shows you the core domain of p53 interacting with DNA. You can see that there is an alpha helix sitting in the major groove and a smaller helix that interacts with the minor groove. So how can we understand the effects of mutations? Well, you can look at a number of different things, and this is a set that we use. We use some other things as well, but these are the main things. Mutations to a proline and from a glycine. Remember that proline and glycine are the unusual structural amino acids. Looking directly at functional residues, so residues that are involved in the function, in this case in binding to DNA. Residues that are involved in protein-protein interactions. Highly conserved residues, presumably they're conserved for a reason. Residues involved in hydrogen bonding and therefore stabilising the structure. Residue clashes, so if we mutated a residue and the replacement clashed with the surrounding part of the protein, well, the protein couldn't fold up in exactly the same way. And similarly, creation of voids. If you had a large amino acid and replaced it by a small one, then if you kept the same conformation of the protein, that could lead to a void in the middle of the protein, and the protein would prefer to change its conformation because having that void there would be unfavourable. So what we do is take this sort of analysis and a few other factors that we look at and feed the results into a random forest machine learning predictor in order to predict pathogenic versus neutral mutations. We've actually also modified the same idea and used it in the prediction of two different phenotypes related to heart disease. So let's just have a look at what I meant by some of those specific analyses. Mutations 2 proline. Remember, the cyclic side chain of proline limits backbone flexibility. The phi angle between the N and the C alpha can't rotate in the same way that the other amino acids can. So here I'm looking at my example of P53 with a Ramachandran plot where we're plotting the phi angle along the x-axis and the psi angle, that's the one between the C alpha and the carbonyl carbon, on the y-axis. Now these squares represent amino acids that are known to have been mutated to proline in P53 from cancer patients. Now the shaded region here is the region which is favourable for prolines. So all of these residues, and these ones out here in the bright red, are positions where this mutation to a proline is going to have to result in some change in the three-dimensional structure. The ones that are in orange are kind of borderline, and the ones that are in yellow are okay for a proline. So this is just highlighting on the structure of P53 where all those mutations to proline occur, and you can see that they're really scattered over the whole structure. What about hydrogen bonds? Well, in this example we've got a hydrogen bond being formed between the OH group of this tyrosine and the ND2 nitrogen on this asparagine. Now obviously if we mutated this tyrosine to something like a valine, then it wouldn't be able to form a hydrogen bond. But what if we mutated it to another asparagine? Would we still be able to form the hydrogen bond? We would have to take into account flexibility of the side chains in order to see whether that interaction could still occur. So those are just a couple of examples of the sorts of structural analysis that we do. Having done those things, as well as making the predictions about whether a mutation is going to be damaging or not, we can also classify the mutations. So functional mutations are making changes to residues that are critical for the protein function. That might be binding to something, it might be an enzyme active site, or whatever. We can also look at fold preventing mutations. So these are ones that really stop the correct fold from being attained. For example, the small to large mutations, which mean that the residue is too bulky and we cannot fit into the folded structure. The third category is really the most interesting because these are destabilizing mutations. So they don't stop you from folding up the protein in the correct way, and they're not affecting the function directly, but the energy of the correctly folded state is raised compared with some misfolded or unfolded state. And it should be possible then to rescue protein function for these destabilizing mutations. Everything in biology is in equilibrium. So proteins exist in an equilibrium between unfolded or misfolded states and the correctly folded state. But normally, of course, this is highly skewed towards the left and the correctly folded form. A destabilizing mutation then is basically skewing this equilibrium towards the unfolded state or other incorrectly folded states. Now, we know this is clearly the case in P53 because one common mutation is seen to be temperature sensitive. And there have even been some experiments done where the body temperature has been lowered during treatment in order to restore P53 function. So, as I said before, conventional chemotherapy and radiotherapy work largely by triggering P53 because there's massive DNA damage as a result of those things. And if your P53 is not working properly, then the treatment is less effective. Consequently, if you can identify the mutations that are temperature sensitive, then you can lower the body temperature and restore the function of P53. The other way of doing it would be with drugs. So if you have a drug that binds only to the correctly folded form, this will mop up that folded P53 and therefore skew this equilibrium. So we would end up then with an equilibrium where the drug is binding to the correctly folded form, and by this kind of law of mass action we would pull more of the P53 into the correctly folded form. Now, Alan Furst, working in Cambridge, has designed small molecules, in fact small peptides, which bind to P53 and confirm this concept. Unfortunately, none of the peptide molecules that he produced had sufficient binding affinity to be useful as drug leads, but at least the concept has been proven. So summarising what we can do with looking at mutations, we can understand the structure, we can understand the local effects of mutations, we can then go on to use machine learning methods to predict whether mutations will be damaging, and we can also predict the global effects of mutations in order to classify them. From that, we hope to be able to go on to design drugs that can rescue function and then give the drugs only to those patients who will benefit from them. Finally, we're going to look at engineering proteins for therapy. Now, here I'm going to talk about antibodies, and some of you will have heard parts of these things before. So an antibody is a Y-shaped molecule consisting of two identical heavy chains shown in blue and yellow and two identical light chains shown in red. Now, the business end of the antibody that we're really interested in from a drug design point of view is this region here, the VH and VL domains that come together to form the FV or variable fragment. And here we're looking at, on the right, the same sort of view of the FV fragment as we have here, but I've highlighted these 6 complementarity determining regions, or CDRs, or hypervariable loops, that form the binding site, and on the left I've rolled this towards you so you can see the binding site consisting of those loops. Now, these 6 loops are really predominant in binding to an antigen. Now, antibodies are really very widely used now as treatments in all sorts of different diseases, but probably the best known application is in cancer. So trastuzumab, also known by its commercial name of Herceptin, binds to HER2, which is an EGF receptor, and that HER2 is often overexpressed in breast cancer, and therefore having an antibody that binds to HER2 can not only help to identify and kill those cancer cells, but also simply stops EGF from interacting with the HER2 and causing those cells to grow and divide. So what I've shown here is the drug Herceptin, and this lady Elaine Barber, who some years ago went to the High Court to be granted permission to have this drug, because at the time it was so expensive that it was being limited in who could get it. Now, as I say, antibody-based drugs are used for all sorts of things now, from high cholesterol to migraine, to things like psoriasis, rheumatoid arthritis, and so on and so on. A small number are used for infections. That's actually relatively rare. But having said that, again with SARS-CoV-2 and COVID-19, there are over 200 antibody-based drugs now in development for treatment of that condition. Now, to use an antibody as a drug, we want to use what's known as a monoclonal antibody. In other words, all of the antibodies in the drug are identical to one another. Now, to do that with human antibodies is somewhat difficult. So in general, we can't generate human monoclonal antibodies in a practical and useful way. There are a number of problems. For example, most of the drug targets that we're interested in are human proteins, and therefore we don't produce antibodies against them. If it were for foreign substances, then there would be ethical problems, obviously, in injecting a patient with some nasty antigen. But that said, a number of these antibodies that are being developed for COVID-19 have actually initially been isolated from recovering patients. So there are some routes now to produce antibodies directly from humans. And there are other techniques, such as using transgenic mice to produce antibodies from humans. But still, a more conventional route is used very widely, and that is to produce what's known as humanized antibodies. So antibody-producing B cells don't reproduce and only live for a few days in culture. So the traditional route of producing a monoclonal antibody is to take one of these antibody-producing cells and fuse it with a myeloma cell line. Now this immortalizes the antibody-producing B cell, and the cell can carry on dividing and producing antibodies. Now, one of the practical problems with producing human monoclonals is that nobody's managed to find a suitable myeloma cell line that we can do this with. Yet it's been done quite easily in mice, rats, rabbits, and all sorts of other species. Now, we also can't use mouse antibodies or other foreign antibodies, because if we inject those into a human host, we get what's known as a human anti-mouse antibody, or more generally, a anti-antibody response against that injected antibody. So this is where this idea of humanization comes in. And what that involves is what's known as CDR grafting, where we take the CDR loops, these 6 hypervariable loops, from something like a mouse antibody, and we graft them onto a human framework. However, that generally doesn't achieve very good binding. So what we need to do is make some additional mutations in the supporting framework from human to mouse in order to obtain good binding. And there are a number of ways of identifying those, all of which rely on protein modelling, bioinformatics, understanding what's going on. So these residues fall into a number of categories. They may be surface residues near the CDRs, because those might also interact with the antigen. They may be residues that pack against the CDRs and define their precise conformation. They may be residues at the interface between the VH and VL domains that influence the packing between those two domains. And there are also other residues that may have an important effect and don't fall into any of those obvious categories. Now, this is a very commercially interesting area. So there's one particular patent known as the Adair patent, where there are 4 residues involved. H6, which packs inwards towards the base of some of the CDRs. H24, which packs inwards towards CDR H1. H50, which is at the base of CDR H2. And also this residue, H23, which is one of these other ones that's difficult to categorise because it actually sticks out onto the surface. This, I should point out, is an antigen interacting with the CDRs. So this is quite a long way away. It's not interacting with CDRs directly. It's not interacting with the antigen directly. So what is it doing? Well, it appears that it interacts with this loop, which is not part of the binding site, maybe modifies its conformation, and that in turn changes the packing against the loops up here. So this is something that wouldn't really be obvious from a simple analysis of the structure or of the sequences. Yet it's known when you change this residue between, for example, a serine and a lysine, that it can have quite a major effect on the binding affinity. So this is just one example of how people do humanization. Other examples involve sequence analysis, looking for residues that are unusual. People also look at models, look at residues that are interacting with the CDRs or near to the CDRs, and use those sorts of changes as a basis for selecting which residues should be mutated in order to achieve good binding. There are other applications of bioinformatics and protein modelling in engineering antibodies as well. So Humira, which is the trade name for adalimumab, is the world's top-selling drug. It's used for treatment of rheumatoid arthritis, and it's generally regarded to be a very good and very safe drug. However, it turns out that more than 25% of patients taking adalimumab develop an immune response against the antibody. And for those who do, the effectiveness of the antibody as a treatment decreases. So why might that be? Adalimumab is, at least in theory, a fully human antibody. It's produced by a technique called phage display. But when we look at the frequencies of the residues in adalimumab, what I've done here is highlight those that are unusual. So we have some on the heavy chain, which are shown in red, some on the light chain, shown in yellow. And although the sequence numbers aren't necessarily that close to each other, these come together to form a patch of unusual residues on the surface. And again, this is not actually that far from this other patch of unusual residues over here. So altogether, this means that we have all these unusual residues on one side of the protein, and this may result in an immune response against these residues. So by using bioinformatics to look at the frequencies of residues, by mapping those then onto a structure, which could be a model or could be a solved structure, we can use this sort of technique in order to look at potential new antibody-based drugs to see whether they may result in an immune response. In summary, bioinformatics and structural analysis has many medical applications. It can be involved in the diagnosis and prediction of disease susceptibility, understanding of disease processes, customized treatment of disease, and engineering of proteins for human therapy. It can be used in diagnosis of disease, prediction of disease risk, prediction of prognosis, customised response to disease, in other words, giving suitable amounts of drug, or indeed giving different drugs depending on an individual's genetic makeup, identifying drug targets in order to lead to new treatments, and engineering of proteins for therapy. However, structure doesn't answer everything as yet. We saw that in that example with the antibody with that particular residue position that you wouldn't have predicted from looking at the structure alone as being a really important position. Finally, here's some further reading, a few mostly fairly old papers but quite key to understanding things like genomics, proteomics, bioinformatics and their applications in medicine and drug design.